Okay, welcome to this week's biomass webinar. We are delighted to have Dr. Erik Sachs of the University of Illinois joining us today, talking about what, what should be a simple question, but never is. What is MXG? And then insight on developing and refining Miscanthus varieties. This could be a whole series just from Erik. So if you're interested in digging into some of these topics in future webinars, not that we would impose too much on Dr. Sachs, but please express your interest in the chat if there are certain topics that you would like to see expanded upon. And with that, Erik, could you go ahead and skip ahead and we'll give some acknowledgements to our funders? So the webinar series is brought to us from by the University of Illinois and multiple institutions within the University of Illinois. For example, the Integrated Bioprocessing Research Laboratory, the Institute for Sustainability, Energy and Environment, and the Institute for Genomic Biology. This is also a collaboration with Iowa State University, particularly the Department of Agronomy. And we are funded by the Department of Energy's Center for Advanced Bioenergy and Bioproducts Innovation, CABI, as well as the USDA's Diverse Corn Belt SAS CAP project. We're also fortunate to have funding from iRegen, which is a group funded by a group of foundations primarily within Illinois. And on the next slide, you can register for the webinar and share with anyone else with the QR code. Then today we're hearing from Dr. Sachs. Next week, we will hear from Dr. Nicholas Boersma of the Iowa State University, talking about some of the long-term trials that we have with Miscanthus in Iowa that help reveal age and fertility dynamics that we can expect from the sterile MXG as it proceeds through at least a 15-year horizon at this point. If you have other speakers that you would like to hear in this webinar series, please also share that in the chat or send it directly with me. There is one slight modification to this agenda. We currently have an opening on March 6th. I have a few people in mind, but Steve Croxton from Drax is going to be in Greece with his wife and thought perhaps he should not do a webinar series while on his anniversary trip. So with that, we will go back to Erik, and we look forward to your presentation. Thank you, Erik.
Sure. Well, thank you. This is a fun topic because it not only gets into questions of, breeding and genetics and nomenclature, but also history. And so I kind of think of it like a whodunit, only over a century maybe or more, and no one got killed as far as I know. So the first question we had was, what is Miscanthus x giganteus, or we abbreviate as MXG? And the simple answer is Miscanthus giganteus is a nothospecies. So, then the next question, thank you for asking, is what is a nothospecies? Right? It just rolls off the tongue, right? Well, nothospecies are derived from crosses between two or more species. And critically, they're defined entirely by their parental species. And this includes the original cross, the F1, any intermediate generations, and any backcross generations. However, they are not defined by their ploidy, just the parental species. And they're not a clone. They're a species with potentially an infinite number of possible genotypes or clones. And I didn't make any of this up. This is all based on standards of the International Code of Nomenclature for Algae, Fungi, and Plants. And taxonomists have been using this code for generations. So we don't have to reinvent the wheel. We're just following the rules of the road for naming critters and plants. So what does the International Code of Nomenclature say about this? It says when all parent taxa are known, a nothotaxon is circumscribed so as to include all the individuals recognizably derived from the crossing of the representatives of the stated parent taxa. F1s, backcrosses, all of that. And there can only be one correct name corresponding to a particular hybrid formula, and that is the earliest legitimate name at the appropriate rank. And anything else is that same hybrid formula is essentially a synonym of the correct name. And so they give a very interesting example here. They talk about Oenothera X. Drawertii. And they say there's two names that were attributed to this. There was a name, oh, okay, yeah. So that's one of them. That's one name. And then the other one was Onothera winei. Now you can imagine in the contest here who's going to lose. Winei. I can't make this stuff up, right? And so both are considered to apply to the hybrid of O. biennis by O. villosa. And it says the types of the two nothospecific names are known to differ by a whole genome complex. What they mean by the types is they mean the type specimen. So when a taxonomist identifies a species, they make a herbarium specimen, and the first one that defines that species, the one to which all others are compared, is called the type specimen. And so they're saying that of these two that were named, one is a higher ploidy than the other, but it doesn't matter. They have the same parents. And therefore, the earliest one, Drawertii, is the correct one. And the other one is just a synonym of it. So what does that mean for us? Well, in 2001, Hodkinson and Renvois defined Miscanthus giganteus properly. And they had noted that the name had been used before, and Griefenduder had described this name, but they didn't do it quite right because, number one, they didn't give a description of this species in Latin as it was supposed to have been, and they didn't provide a type specimen for it. Now, that's not according to the rules of International Code of Botanical Nomenclature, so these taxonomists fixed this problem, and they defined it in Latin, and they made a type specimen. And so they said after this, and this is unfortunate language because it caused a little bit of confusion. They said, we have shown it to be an allotriploid 2n equals 3x equals 57. And with genomes from Sinensis and Sacroflorus. When they said it, they were referring to the designated type specimen, not to the species. And remember that all of this here in English, this is not the definition of giganteus, even though we can read it and understand it clearly. The real definition is down below here in Latin. And it says giganteus is a hybrid of Miscanthus sinensis and Sacroflorus. End sentence right there. That's it. That is the definition of this nothospecies. Not the ploidy, but the species. And they made a type specimen at Kew Botanical Gardens, and they called it 1993-1780. And thankfully, Hodkinson saved a live plant of this in his garden, and he sent me a piece of it. So we have it.
Oh,
Yeah. Isn't that cool?
What's the name of it, Eric? Does it have a special name?
I call it 1993-1780. I don't rename it. And so in my papers, when you read, about MXG 1993-1780, we kind of say it comes from this and we refer to this paper, but this is it. Okay, MXG, the standard clone. I feel like it should be a movie, maybe an epic, okay? So in 1935, Axel Olsen, he was a nurseryman in Denmark. He imported this Miscanthus clone, this very same clone, from Yokohama, Japan. And that got distributed throughout Europe and eventually made its way to the United States. Olsen didn't likely travel to Japan, but he ordered this plant probably from a catalog and had it shipped to him. He was also known to be the founder of a geographical garden, a botanical garden in the city of Kolding in 1925. I'll show you a photo of that in a second. It still exists today. And later, Carl Forster, who was a very famous and influential nurseryman and thinker about plants, he really instituted a second wave of ornamental grasses being used in the landscape in Europe and North America. He called it Sinensis giganteus. That's where the giganteus word came from. I very much doubt when it got from Japan to Denmark, it came with that name. I think I know what name it came with, and it didn't come with this name. And it subsequently got renamed X. giganteus by Griefenduder, right? Because it really wasn't a sinensis. So we don't know how it came to the US, but what we do know is that the clone that we have in Illinois came from the Chicago Botanic Garden. And so we named it Illinois. And I think that was that Tom Voigt who did that?
Tom brought it here. And then when we started needing to write about it, that's the name. I think Steve Long is the one who put our stamp on it.
Okay, so anyway, that's how it got the name Illinois. And that's kind of what happens. And I will tell you, it is genetically identical to the standard clone, to the type specimen. I found no differences between it and the type specimen in Europe. And so this is an example of what happens when people move clones around the world over many generations, many years, is it tends to go to a new place and get a new name, but it's really the same exact thing. Here's the geographical garden in modern times in Kolding. It was meant to show plants from Japan, Korea, North America, in Denmark. Very pretty place. And here is a picture of a catalog of the Yokohama Nursery Company from 1898, and showing for sale, for some amount of gold, $1 gold or $1.50 gold, you can get some really strong clumps shipped to you in 1898 of, for example, Miscanthus sacrifloris, which the Japanese name is Ogi. Remember that, because I'm going to come back to it in a moment. Here's what they called Eulalia japonica zebrina. This is what we would call today Miscanthus sinensis zebrinus. In their time, in the English-speaking world, we would have called this Eulalia japonica. So the names have changed a little bit, but these are the same plants. They called sinensis in Japan Suzuki. Keep those two names in mind. Suzuki for sinensis, Ogi for sacrifloris. The Yokohama Nursery Company was a very famous and influential company for ornamental horticulture in Europe and North America. It was founded in 1890. And that same year, they opened an office in San Francisco. In 1893, they presented the first bonsai exhibit to Westerners at the Columbia Exposition in our very own Chicago. And they exhibited some 40 maple varieties, 150 iris varieties, and the first Japanese garden constructed in America. They opened a branch in 1898 in New York and in 1907 in London. And in 1912, they shipped the cherry trees that Washington, D.C. is so famous for, the first batch. The company's still in existence today. It's very different now, obviously. But they made these beautiful color catalogs in English and sent them all over North America and Europe. And so even though our records of the giganteus clone going to Axel Olsen say he got it from Yokohama, it doesn't say what nursery. I would bet they got it from this nursery, right? And so this, just so, for you guys to remember, this is on one side of Tokyo Bay, right? So this is next to Tokyo. And Yokohama was one of the first major ports open to Western trade, right? Admiral Perry landed there and said, okay, you got to open up trade. Not a great history. So when we looked using genetic markers, for example, where our sinensis came from, the ornamental cultivars, the naturalized cultivars, we could determine that the ornamental cultivars all were closely related to, and therefore came from southwestern Japan and right around Yokohama. Not too surprising. And the earliest record that I can find of Miscanthus being brought to the US is from the 1870s, where Zebrinus and Varieties were imported from Japan by a Mr. Thomas Hogg, a nurseryman from New York City. This is a record of that introduction of Zebrinus from 1876 in the American Agriculturalist. It's so great. We got all these things scanned in and on the web these days, makes it a lot easier. And here it is, they called it Eulalia japonica, right? But we know what it is. So there's another possible name for Miscanthus giganteus, and that is Miscanthus ogiformis, the form of Ogi. It looks a lot like Sacrifloris, but it isn't. And this was described by a taxonomist called Honda in Japan in 1939 in the Botanical Magazine. The type specimen, which is in the University of Tokyo Herbarium, was made in 1937. Remember, Olsen got his plant in 1935. And he says that the plant is endemic, and the type specimen is from Kyushu, from province Higo, which is shown right here. Very subtropical. They grow sugarcane around here. And Higo doesn't exist as a province anymore. It's now part of a larger prefecture. But anyway, that's where it's from, a very specific place. And he provided a nice description in Latin, which is great. He followed the rules. He made a type specimen. He never said in Latin that it is a hybrid of Sacroflorus and sinensis, which is a little bit problematic for us. But we know that he knew it was, but he said the common name in Japanese is Ogi Suzuki. Ogi is Sacroflorus Suzuki sinensis. He knew it. Now, some of you may know Ryan Stewart, who used to work at U of I and now works at Brigham Young University. He actually went to Tokyo University Herbarium and got them to give him a little piece of leaf from this herbarium type specimen. And all of us have been too scared to see if we can get any DNA out of it, you know, because it's like almost 100 years old, right? And but if we could probably see whether it's the same genotype, the same Sacroflorus bisinensis, you know, is it really giganteus? And also, is it the same genotype as the main one? If I were a betting person, I would bet it is the same one that was in the Yokohama Nursery Company across the bay, and the one that probably got sent to Axel Olsen. It's just too much of a coincidence that in the 1930s there could have been multiple ones of these floating around. So Sacroflorus and sinensis are the parents. What I'm showing you here are two maps of where Sacroflorus and sinensis comes from. On the left side is Sacroflorus, on the right side is sinensis. Each dot represents a collection at that particular location with GPS coordinates. And the circles here, these are all, circles are diploids. The squares are tetraploids. So a diploid means it's got one set of chromosomes from mom and one from dad. The tetraploids have two from mom and two from dad. And there's some triangles in here, like over here. Those are triploids. And the colors represent genetic groups. And so there's 6 different genetic groups of Sacroflorus, 3 diploid, 3 tetraploid, 2 from Japan tetraploid, and 1 from Korea, other side of the Yellow Sea, Shandong, as well as some up here in Russia. And there are 7 genetic groups of sinensis, 3 in Japan, South, Central, and North, a whole Korean group here, a whole bunch of tropical ones down there, and then here's Sichuan Basin and Yangtze Kingling. Anyway, so there's a lot of diversity, and the fact that there's such an overlap between Sacroflorus and sinensis means there was a lot of opportunity for these hybrids to occur. And in fact, triploid and tetraploid hybrids are pretty common in southern Japan and in southern parts of Korea. And diploid hybrids are common in the mainland, and especially in the north. This was a study where we looked at this. And so what you see down here is the proportion of Sacroflorus ancestry, nuclear markers, and then latitude. And here we have sinensis. These are pure sinensis, these red circles. And then these here in blue are Sacroflorus. And the ones that are 100% ancestry are pure Sacroflorus. Now, if it has no fill, then it's diploid. So like the sinensis are all diploid. If it has a little X mark in it, it's triploid, like our giganteus Illinois. And if it's filled, it is a tetraploid. So the haplotype, so the female, or so the plastid genetics comes from the female only. And so here you can see some of these hybrids, some of these triploid MXGs have Sacroflorus as the female, and some have sinensis as the female. The cross can go either way. And you can also see there's a whole bunch that are tetraploids, and there's a whole range of gradation of how much ancestry from Sacroflorus they have. This is what we call hybrid swarm. All of these are giganteus. All these are giganteus. They're all natural giganteus.
Did you say hybrid swarm?
I call it a hybrid swarm when you have these 2 different types and they're all mixing together. What you have is an introgression of sinensis genes into tetraploid Sacroflorus. What happens is you get an unreduced gamete on the sinensis side. So not half of 2 is 2. And then half of 4 is 2. And 2 and 2 is 4. And that's how you get tetraploid. And you start intermating with the tetraploid other ones. And so that's how you get this. This was unknown without the markers, right? All we knew is we have these triploid hybrids, but no one really knew there was this whole gradation going on in terms of genetics. But the ploidies are separate. So MXG ploidy diversity is kind of large. We can have diploid giganteus, we can have triploid giganteus, tetraploid, pentaploids, hexaploids. We have all of those in our collection. Here, and so the way this happens, if you have a diploid crossed to a diploid, you usually get a diploid back, right? Reduction division for meiosis. If you have a diploid crossed to a tetraploid, you usually get a triploid back. But sometimes these diploids don't have reduced gametes. You get unreduced gametes, and so then you get a 4x back, like I just said. And then if you had a 2x by 4x, you get a pentaploid. And if you double the chromosome number of a triploid with like colchicine, you can get a hexaploid. We have those. So that can be really confusing. If you just say MXG, you don't know what ploidy it is. So what we recommend is that we include the ploidy after the species name when we describe our varieties. So for example, Giganteus 2X 18UI4910, or Giganteus 3X Illinois, but if we got a chromosome doubled one, it's 6X Illinois. Or 4X 17UI66R10, right? So these are some things in our collection. So we looked at markers at all these different giganteus that were out there. This was Kasha Gloaka who did this. And what we found was, so these lines represent how different things are from each other. And so what we have is a bunch of really old, what I call legacy giganteus. We have the 1993, 1780. We got a bunch of different Illinois. We've got Hortico. We've got something from Georgia. We got the Danish one. Something from Wisconsin, the chromosome doubled ones, a German and Polish one. And these were with SSR mark. They're all the same. What little bit of difference we found here is marker error. Every marker system you use will have some error rate. And the only way to distinguish error rate from real differences is to do technical reps. You basically put the markers on the same exact genotype, a different DNA extraction of the same individual, and see if you get any variation. That's the error rate. Every system has an error rate, so you have to define it. So then we have some crosses here that are true giganteus crosses. And you see these 10UI32s, -1, 2, 3, 4. Those are full sibs of a cross that I made. So they have the same mom and the same dad. They're brothers and sisters, both the same parents. See how different they are from each other relative to all of this? No. These are true differences among very closely related individuals. They're full sibs. And then these Ogi 79, 80, these and 63, these were collected from seed in the wild in southern Japan. Those are true giganteus. And Nagara is another giganteus. It's a cross that was made by Martin Deuter, a tin plant. So that's kind of what's going on there. And then the other really strange thing that we found was that we have about 100 ornamental cultivars that are sold in the US and Europe that are called Miscanthus sinensis. And we found that with the markers, about half of them are really Miscanthus giganteus 2X purpuricens backcrossed to sinensis 1 or 2 times. So these are all really giganteus, but they're all sold as sinensis, which is what they mostly look like. And in the 20th century, the key breeders of ornamental Miscanthus were from Germany. They were Karl Forster, I already talked about, Ernst Pagels. And so they were trying to breed in earlier flowering time and also greater winter hardiness, right? Because the Japanese materials really maritime, warm, not very cold hardy. They probably wanted to get them at later flowering. They probably wanted them to be earlier for the ornamental. They wanted them to be shorter, and they wanted them to be more cold hardy in the wintertime. And so they used, whoever did this, probably Pagels, used this as the source of that, and then backcross to sinensis. So that's
Eric,
kind of the food. Yeah.
did you say what the y-axis is on that figure? Is that the amount of Sacroflorus again?
So the color coding are the different genetic groups. So we have blue for Sacroflorus, yellow for South Japan, North Japan, green is Central Japan, and red is a sinensis from mainland North China, which is where the purpuricens is coming from. So only these hybrids have any sinensis from mainland Asia in them, and it's coming from this purpuricens.
Thank you.
Yeah. So we use the same color coding with all our publications, so we try to keep it pretty pretty consistent. Um, so, why does this all matter? Well, so here's 1993-78 or Illinois. This was 2013. This is for, first spring after a very cold winter when it got down to minus 6.2 Celsius at 10 centimeter soil depth. And in Dixon Springs, they were fine, southern Illinois. But in Urbana, we killed a lot of them, and a lot of them were really badly damaged. But Nagara, which, by the way, is called Nagara because the parents were from the Nagara River in Gifu in central Japan, a little bit further north. It's more winter hardy, right? And then, some other things. Here's one of those crosses I made, a little bit better, and this is another one, not so good. So it varies depending on where the parents are from. And so we've done some studies looking at freeze tolerance of dormant plants, of Sacroflorus up here at the top and Sinensis here at the bottom. This is what we call the LT50, or the lethal temperature that kills 50% of the population. And you can see some of them, we don't kill half of them until we get down to minus 17 or 18. These are like the Russian ones, or Robustus, which is also from the same area, the Amur River region. And others, which are tropical or subtropical in origin, are killed at minus 2 or minus 3 pretty easily. You can see here, again, Nagara is more cold hardy than Illinois. And we have some variability, but not as we haven't used as much of the variation as we actually have to get the kind of winter hardiness we'd really like to see. But we have that variation. And so we've looked at some ways of looking at soil temperature and relating, what the median annual minimum soil temperature is over the last 30 years, or the 25th percentile, so like the lowest part of what's typical, or the lowest annual minimum temperature in the last 30 years. And you can see it can be pretty low for some of these regions, Iowa, central Illinois, etc. And so then we asked, knowing what the LT50 is from our freezer tests for Giganteus, Illinois, or Giganteus Nagara, what kind of survival would we expect to get? And in a typical year, we'd have pretty good survival in the Midwest, in the lower Midwest anyway. But in the worst case scenario, we're going to see a lot of winter kill. And we do indeed see that. And Nagara is a little bit better than Illinois, which bears out. But look at what we could do with Robustus. This is the potential of what we could create, right? This is Sacroflorus, not a Giganteus. It's essentially going to be unharmed throughout our target growing area here in the Midwest. So we can do better. That's my point there. So what's coming down the road, right? Here's 2 new promising triploid Giganteus selections for the Midwest that we've trialed in 4-row medium-sized block trials at multiple locations. One that I'm especially excited about is this 12UI17. We're looking at dry weight yields of megagrams per hectare in year 2, 3, and 4 in Urbana, and year 2 and 3 in Letts, Iowa. Thank you, Steve Schoenberg, for this, and Emily for helping to plant it. And notice in 4 out of the 5 year location combinations, it is our top yielding entry. 4 out of the 5. And it matches pretty well. And then here's another one that's doing pretty good, the 13UI1912. Nagara beats out Illinois in year 2, which is pretty typical. Illinois is slow to get going. In year 3, Illinois beats it out. Year 4, I think, was a drought year, so not as good. In Iowa, year 2, again, Nagara beats out Illinois, and then Illinois beats out Nagara, but still 12UI17 beating out all of them. Well, so this is how we harvested those trials with a 4-row harvester, just so you get a sense of it. At least the ones in Illinois. I think in Iowa we did it by hand. And then this is what those plants look like. So here's 13UI912. There's a meter stick here. And this is year 3. And here's 12UI17. You're probably going, hey, wait up, Erik. That's 12UI17. It's really short. Well, it isn't really short. It's just not tall. How can it yield so much? Well, if you look really closely, it has a boatload of stems. It's got a lot of stems. They're thin stems, but there are a lot of them. And that's how it makes up that yield. And it doesn't lodge because it's not too tall. I don't care what it looks like as long as it yields. Here's the Illinois clone, much taller, but look at all that space between the stems. No space between the stems. It's much more of a clumper. This one's more of a spreader.
Erik,
here's the Gara, a little bit prone to lodging. Yep.
Yeah, Erik, the Iowa one year the first year we harvested, or second year, I guess, we harvested with the 4-row
oh, okay.
chopper.
Cool.
Your team came over and
Yeah, that's a big job to bring it over. But yeah, cool. So that's pretty consistent then. So anyway, these are some things that are coming along. Here's two more things that are coming along I'm pretty excited about. This was taken in Alabama at Alabama ANU Experiment Station this summer. I took these photos. Here's the Illinois clone at the end of August flowering, right, because of day length. But right next to it is a selection that isn't yet flowering. It's not even flagging, and it's already taller vegetatively than the Illinois clone. So this is better adapted for the South. Here's another clone that has flag leaves at the same time, also very tall, also very upright, not flowering. So this is later than Illinois, which should be advantageous for biomass yield in the South. So these are some of the things that are coming along. And this is my last slide, just saying Illinois is the land of Lincoln and also the land of Miscanthus research and development. This is taken in the park right across from the high school here in Urbana. I just couldn't resist. Happy to take any questions.
I know I'm not a normal audience member, but I'm going to stand up.
Oh, thank you, thank you, thank you. Very kind.
Yeah, I feel sorry for people watching the recording since I'm just grinning in this whole thing here, but that was amazing. I think we have, let's see, I don't know that we have a lot of questions written in the Slack, but people can put them there. I know we probably do have a lot of questions in general. I'm going to kick it off if that's okay. A very practical question. You told us when we're writing the name, you gave us some examples of how
Yeah.
to do that. Do we need to put the chromosome number in there or just the ploidy?
I would, if it's a true whole genome set, n equals 19. So if it's a multiple of that, then the ploidy should tell you how many chromosomes you have. If you have an aneuploid, well, then I think you need to say something about that. But that's not so common.
Okay. You would let us know if we did, because we'd be working with your materials.
Well, in order to figure that out, you actually have to do chromosome counts. Flow cytometry won't get you that accurate an understanding of it. So if they're out there, we probably are not aware of it, but those would be probably pretty infertile.
Great.
Yeah.
That was my second question, is the robustus type with all the cold tolerance. Tell us your thinking about how that gets used to incorporate the cold tolerance given the fertility.
So robustus is potentially fertile. We've made crosses with it. And most of those Russian Sacroflorus are fertile, male fertile and female fertile. It happens to be that the robustus selection, though, which was made by Forrester, actually, in the 1950s, he made that selection. It's male sterile as far as I can tell. And so we've always used it as the female parent. It is in the horticulture trade still, so you can get it, but it's not going to produce any pollen, and it doesn't really have anybody to cross with when it's out there.
That's very helpful. I remember seeing when I first started at Iowa State that purpurescence had been in the botanic garden there back when it was the State College of Iowa, and they removed it because it kept spreading. So this is just such a cool historical arc with these that we could probably have even regional stories that go with it.
Oh my gosh, yes.
All right, other questions for Erik now?
Erik, how does the Illinois clone differ from the clone that we plant primarily for commercial purposes now?
Oh, the Freedom clone.
Yeah.
So that's an interesting story. The short answer is it doesn't genetically, as far as we can tell, within our ability to detect. So what does that hedge mean? That means we threw a huge amount of markers on it, including sequencing markers, and we could find absolutely no difference between it and the standard clone and the type specimen. Now, it could very well be if there were a point mutation, a single change in one nucleotide, we would not detect that probably. It'd be like a needle in a haystack. You'd have to have very deep whole genome sequencing to even have a chance of finding it. And you would often question whether it was real or a sequencing error. So That's a possibility. I won't exclude that possibility. But what I can tell you is for certain, it came from the same original seedling. So when that seedling got produced in the wild of Japan and then got moved around, it's possible that some of those vegetative divisions accumulated a small mutation here or there. Those are called sports in horticulture, right? I won't deny that that could be a possibility. I haven't detected it. Um, the story that I understand from Brian is that he originally got it labeled florigelus, and then they realized it was really a giganteus. I think they got it from the National Plant Germplasm System, if I'm not mistaken. And he said after selecting it several times vegetatively, he had one type that was better than the others. In my hands, in my tests in the field, I've never seen it perform differently than Illinois clone. I don't think there's any difference between it. It's patented, which kind of really bothers me because it's identical to the freely available clone. So how are we going to defend? How would anybody defend that patent? Or are they going to claim to own everything that's freely available and has been for 90 years? I don't know what you do with that.
That was the question I was going to ask. Because what the implications are that many of the supposed varieties are identical, how you trademark anything, and whether or not the ones that are trademarked are even valid.
It was trademarked and now it's patented. That's even worse.
Do you think it's valid? I mean, is there
Well, it's valid until you challenge it in court, but who's going to spend that money?
Right. Someone that wants to sell Miscanthus, probably, honestly. I don't mean,
and has a lot of money. There aren't too many of those people around. Yeah. So they're all the same.
Yet, Eric, yet.
Well, those are
people who have money and want to sell Miscanthus. Let's hope it's yet. Can you talk a little bit about your breeding program and how you would think about releasing varieties? How does this go back to the land grant system, or does it? We just explain a little bit about how your work gets into the commercial sphere.
Well, not really much different than most other cultivars that would be produced in a land grant system these days. So these days, most universities will take something that has commercial value and either they'll protect it through patents or contracts or both, and they'll license it. And they'll either license it exclusively or non-exclusively. I'm of the feeling that we should already have such a small marketplace, we should probably keep it fairly open. So I'd prefer to see things marketed non-exclusively. I'm more cautious than maybe some. I want to make sure that when we release something out there, it's not going to be a disaster in the farmer's hands, because if we burn them once, they're not going to come back and believe us later. And I've seen this in other crops, so I'm very cautious. On the other hand, it takes a long time to breed these things. And so I know there's a lot of demand out there. But we're starting, we're going to send, we just signed MTA with Travis. We're going to send a bunch of materials that are promising to be propagated there. And if at some point he wants to license those, he can try to negotiate, he can enter into a negotiation with UIUC for that. But having spoken with Travis quite a lot, I don't know if he's on here or not, but they've invested a lot in this Freedom clone and getting that all propagated up, and so that's going to be a barrier for new varieties within his program getting into the marketplace, at least in the short term. So that is a concern. In Europe, they're moving a little more quickly. They've also been at it a little bit longer.
I'm just going to say, for future watchers of this video, Travis is Travis Hedrick of Agrotech, North America's largest Miscanthus supplier. And Travis will be giving an upcoming webinar about this in the future. And also, I think one of the things that we'd love to hear from you as well is, can you just comment on invasive potential? I know that comes up a lot, and particularly how invasive potential might relate back to the naming that you discussed.
Oh yeah, so that's very important. So actually, all of the ones that are an odd ploidy, like 3X or 5X, these are essentially sterile. they're not going to produce viable seed at any rate that could be of any concern. They might very, very, very rarely produce a few viable seed. But those will not only be odd-ploidy, they'll be aneuploid. They'll also be highly sterile. So when you think about it from a population perspective, if the population is going to be invasive and weedy, it's got to grow. And so if you end up, so there, you look at probability. This is how population genetics looks at this sort of thing, right? So if you have a low probability event, so the probability of producing a viable seed from an odd-ploidy line is very low. Okay, now the probability of any one viable seed surviving somewhere and growing to adulthood is also very low. That's why Miscanthus fertile ones produce lots of viable seed, like thousands and thousands, very tiny. And then the probability that if we did have those two unlikely events occur together and it grew up, then it would have to also produce viable seed. Very, very low probability because it's also pretty sterile. And there's no way a population can grow like that. These are dead ends for population growth. They can't do it. They're going to stay where you plant them. Now, the fertile ones, the even number ploides, those can produce viable seed. Now, remember, Miscanthus is self-incompatible. So a block of all the same genotype, right? That's not going to produce much in the way of viable seed either because it's self-incompatible. It's a leaky system and might produce a few, but again, very low probability. But if it had something else to cross with, some pollen came in from somewhere else, let's say an ornamental planting, then you could get viable seed. And those could, the problem with the Miscanthus right now is that those seed would blow away in the wind and could potentially start up somewhere else because Miscanthus isn't a properly domesticated plant. But in the future, we will have properly domesticated Miscanthus where the seed does not blow away in the wind. And we will eventually have fertile, true ploidy Miscanthus that we can plant directly from seed. Now, in Europe, they're not worried about invasiveness like this, like we are here in US, and they're already planting seeded varieties from seed. Now, their environment is a little bit more stressful and short season, and so they're actually starting those seeds in trays and then transplanting them. But I believe here in where we have more continental climate, we have a much longer, warmer season than, let's say, UK or Germany, we could probably direct seed into the field like we do switchgrass. That's the future, far future, but maybe not that far.
Yeah, give us a year range on that. 5, 10?
Hard to get. Boy. To farmers' fields, 10 to 20.
Okay, thanks. So I think the reason I was asking that in part is we are, you have a support team around you who is helping to ask these questions about how these crops work in the environment. You test this in multiple locations. So the odds of you being surprised are negated, essentially. Okay.
Yeah, we test a lot and for a long time.
Other questions for Erik? I know we're up against our normal end time, but we have time for a couple more.
I'm just curious what the ploidy is of the Miscanthus sold by the Michigan company that advertises a lot. I see it that they sell for deer refuge and wildlife refuge.
There is one company that I also know sells for hunters, basically, to make blinds for deer, etc. And that's just the same Freedom or Giganteus Illinois clone that everyone else has been using.
There was some Nagara that was sold in little refrigerators at some wildlife outfitters.
Okay.
So that's in some states, but again, it's Nagara, so it's still a triploid clone. And there is a thriving community on, if you go to whitetail.com, for example, you can find a lot of information. So there's a thriving trade in the hunting community to use versions for blinds, but I have not seen a great amount of detail on the origins of that. It's got to all be from the same place.
I guess my other question, if there's time, is how long until you would look to license 007?
So that's a good question. What I'd like to do now is propagate it up even further and put it out into larger plots at more locations. I'd like to have some half-acre to 2-acre plots of this and just see how it compares side by side with the standard clone, and then decide, is it consistent? Are we still happy with it? And then try to license it from there. That's going to take a bit of work to do, and money. And the breeding program here at Illinois really has almost entirely been on the side. There's been very little direct money just for the breeding. Most of our funding is for basic science research, so the breeding comes as a sort of on the side. And so that's not it's not the most efficient or fast way to do it. But, we're sort of a chicken and egg scenario here where we don't have a lot of industry with a lot of money to support a breeding program. And we need new varieties if we're going to have a bigger industry. And so, there we are. We're trying to prime the pump. But, if someone came to me today and said, oh, I'd like to license this, I'd certainly entertain a conversation, but with some caveats that, we need to be careful.
Erik, your new varieties, or what's the right terminology, I guess?
Collections, sure.
Your new collections.
Selections.
I think you showed the figure with all the diversity, the genetic differences between the
Yeah.
collection. Yours are different enough that you can detect it, right?
Oh,
Pretty
yeah.
quickly.
Yeah.
Yours are easily licensable, right?
Oh,
Versus
yeah.
what's out in the wild right now.
They're all different, and they're all different from the standard clones and the current varieties. Yeah. I mean, we've made the crosses. We know the parents, so.
Yeah. Also, if I was to make a pilgrimage to the original herbarium specimen, would you be able to show me around over there?
Oh, the plant that we have here, or the one that's in Tokyo?
Yeah, the Illinois pilgrimage first. I feel like there's
Same.
different levels of pilgrimages. We could
I believe that we have it as just a pot well, there's a pot in the greenhouse, and I think we have a plant of it in the field that we keep different genotypes of giganteus around just for future use. Yeah, sure.
Yeah, I'm gonna make a pilgrimage to Illinois to see the herbarium specimen, and then eventually to the Japanese, what'd you say, University of Tokyo, right?
University of Tokyo herbarium.
Yeah, I'm gonna have to go see that. That's pretty cool.
Yeah, I need to do that too. I've not actually seen it with my own eyes. And if anybody knows of someone who is really expert at ancient, at archaeological kind of DNA extraction from plants, we really need an expert to do this.
Well, I think this is a great place to pause the conversation today. Again, reach out to me with additional topics that you'd like to hear Erik explore or dive deeper on some of this. But for now, let's go ahead and please let's thank Erik one more time. That was amazing.
And thanks for your interest.
I will say, if you would like to support the breeding program, that can be done through the University of Illinois Foundation, and I can help facilitate that. Erik can help facilitate that. Unlike many other crops, there is no USDA breeding program for Miscanthus. So as Erik said, it's being done privately, and he subsidizes it through his other research grants. So if you're thinking of an endowment or you know someone who is, this is an excellent resource for the future of energy in this country, or biomass. That's for the YouTube watchers too. I know not everyone on this call is so wealthy.
Bill Gates, this is for you.
Right. Get in early. Okay, well, thanks so much, and we'll see you back next week for Dr. Nick Boersma to tell us about Miscanthus, Miscanthus giganteus Illinois, the triploid clone, as it ages in Iowa. So thank you all very much. Have a great day.